who have heard about this problem, which is very timely today, as you will see. I have no disclosures. I have to confess it's very complicated. The science is complicated, but what makes it more complicated, and I think everybody knows, is the politics, the influence of pharmaceutical industry, and people's beliefs in all kinds of things that have nothing to do with reality. So the summary, I'm going to review the virology of SARS-CoV-2. I'm going to talk about the stages of COVID-19. I'm going to highlight the immune dysfunction and thrombophilia in COVID-19 and discuss an approach to each phase. So just to begin, this is not the flu. People still think this is the flu. So you can see the mortality in every age group in the US compared to flu, significantly greater than the flu. And as you'll see, as a disease, it doesn't resemble the flu. I need to highlight this slide because we all in really big trouble. As of yesterday, there were 70,000 new cases of COVID, and it looks like the graph is going up. So we are in for a bad, bad winter. So where did this all come from? So it seems like the bat, so SARS, MERS, and CoV-2 seems like the origin was the bat. SARS-CoV-1 went through the civet cat, MERS went through a camel, and the intermediate host in SARS-CoV-2 we really don't know. So this is the virus. It's a 0.1 micron enveloped RNA virus. It has a surface spike glycoprotein, an E protein, a membrane protein, and it has a envelope, has a nuclear protein, and a single RNA genome. And as you'll see, it just has one chromosome, but it seems to have outfoxed us humans who have at least 46, or at least some of us. So how does the virus get in? So SARS-CoV-2 binds to the ACE2 receptor. And you'll see the ACE2 receptor is highly expressed in the ciliated epithelium in your nasopharynx and in the lung. Then there is a protease temporis 2, which cleaves the spike protein, exposing a site which allows the spike protein to fuse with the membrane, and therefore then you get injection or whatever of this RNA. So this is the RNA of this virus. It's about 30 kilobases. It's quite an intriguing molecule. So it codes for 28 proteins, only 28 proteins, of which 16 are called non-structural proteins, 4 are structural proteins, and 8 are accessory proteins. One of the major proteins is called this open reading frame 1, which is a polyprotein. That polyprotein then gets broken down by proteases into the non-specific proteases. So how does PCR work? It's really important because I don't think people understand when you get a PCR test, what is it actually testing? So what it's looking for is a tiny nucleotide sequence in the virus's genome, a tiny sequence. So it's not looking at the whole virus and it's not looking at live virus. It's looking at a very short nucleotide sequence. In the US, the commonest sequence is the N protein, part of the N protein. In many European PCRs target the open reading frame 1 RNA-dependent RNA polymerase. So this becomes important, as you will see later. So what happens, as I said, is this virus codes a whole bunch of non-structural proteins that are very, very devious and smart. So for example, non-structural protein 1 inhibits host protein synthesis. So what it does, it gets into the cleavage site of the ribosome and it prevents translation of human RNA. It switches off human RNA synthesis, but it's devious enough that it allows the ribosomes to transcribe viral proteins. It has a protease, an RNA-dependent RNA polymerase. It blocks interferon. It inactivates natural killer cells. It downregulates MCH1. So you can see what the silly virus is doing is downregulating the host immune response, disrupts autophagy. It has another unique feature, actually. It can proofread the RNA to see if there are mistakes. And one of the mistakes it picks up is remdesivir, and it has an endonuclease which cuts it out of the RNA. So this is a very smart virus. So this is just the life cycle of the virus. You can see it gets injected, the RNA gets transcribed, it produces the polyprotein, it produces these non-structural proteins. And then what happens is the virus gets protected in what's called a replication transcription complex, which is a double membrane vesicle in which the virus replicates like a little factory. And these non-structural proteins protect this double vesicle from autophagy. So it provides a protective factory for this virus, and it replicates like a factory. Then we have release of subgenomic RNA, genomic RNA. It then gets all collected together and gets budded out. So this is what it looks like in the cell, these replication transcription complexes, which is part of the Golgi apparatus, and it provides a protective environment for this virus to replicate. So how is it transmitted? And this obviously is a really important question. So the most important is droplet spread. So these are droplets greater than 5 microns and go a distance of 6 feet. So this is really important because everybody needs to know this. There is some evidence of aerosol spread, so this is greater than 6 feet and smaller particles, but it appears to be less dominant. And then there's the question of formites and environmental spread, which really seems to be insignificant. So the most important is droplet spread, and this becomes important in controlling this pandemic. So this is the difference between droplets and aerosol. So airborne can travel quite distance. Droplets go about 6 feet. And like much of this pandemic, there's been enormous confusion about what this virus does. You can see the CDC are flipping backwards and forwards on this issue. Just adds to the confusion. So what happens is you inhale this virus into your nasopharynx. The nasopharynx is a really protective site. You have ciliated epithelium with ACE receptors. The virus replicates in a protected way, creating millions and trillions of virons. So somebody just speaking, if you are infected with SARS-CoV-2 and you just speak, you don't have to cough or sneeze, you exhale about 1 million virons per hour. That's how infectious you are. There are some people then will aspirate the nasopharyngeal contents into the lung, and you get the lung involvement. So this is kind of important because folks don't understand this. So the ACE expression and infectivity is highest in the nasopharynx, goes down as you go down. So this virus replicates most aggressively in the nasopharynx. And this will become evident why this is so important. So the nasopharynx is where this replicating factory is. It has the most ACE2 expression and the highest viral load. So this is a very pesky virus, and this is why it is transmitted so readily. So this is your peak viral load. So in blue is SARS-CoV-2, in red is SARS-1, and in green is MERS. So you can see that SARS-CoV-2 has the highest viral load and is most infectious prior to the onset of symptoms. So prior to the onset of symptoms and in asymptomatic people, you have this enormous viral load. And these people then excrete gazillion copies of this virus. Unlike SARS, which then becomes infectious only when people are symptomatic. So the issue is that the most infectious people are those who don't know they have the virus. And this is critical in understanding this disease. So what can we do? So what can we do? Every single person in this country needs to wear a mask. And masks protect the wearer as well as the rest of the community. So if you wear a mask, it reduces the inoculum size so that you're much more likely to have an asymptomatic or no infection at all. So it's absolutely critical to break the cycle. Everyone wears a mask. And you can see what happened in New York City. The most effective way of combating the outbreak in New York City was a face covering. It's a simple strategy, and people get used to wearing a mask. What we need to understand next is that there are various stages of COVID. And it's very important because the treatment differs dramatically. So you have your incubation phase, which is about 5 days. It can be up to 14 days. Some people will then go on to the symptomatic phase. About 20% of people are asymptomatic. People then go into the symptomatic phase with fever, cough, headache, and malaise. As I said before, viral replication is greatest before the onset of symptoms. Once you become symptomatic, you get rapid decrease in viral replication. You then go to the pulmonary phase. As you can see, the pulmonary phase, there's no live virus. Really important. There's no live virus. What happens is you have, because you've had so much virus replicating, you have a viral graveyard of viral body parts. You have viral body parts, and the host responds to these viral parts. And as you'll see, it's not the virus that kills the host, it's the host immune response to these dead viruses which kills the host. So you have this profound immune dysregulation, T-cell dysfunction, cytokine storm. So just to go through the stages again, so day 1 here is onset of symptoms. You have fever for about 12 days. So the top column are in people who survive. The bottom hatched column is people who die. So fever about 12 days, cough about 18 days. Then you have this window, and this becomes really important. So some people actually think they're getting better. So on about day 6 or 7 or 8, people develop respiratory symptoms. So you have this window. And what then happens is you get shortness of breath. As we know, some patients actually may not be short of breath when they have pulmonary involvement. The so-called happy hypoxics, but really it's silent hypoxemia. So you have lung involvement on about day 7 of symptoms. This is really important. Then you go along short of breath for 2 or 3 days. You then line up on the ICU. You're there for 2 or 3 days. You land up on a ventilator, and then you're dead. So this is what it looks like in the lung. It causes an organizing pneumonia. This is not ARDS, which they treated for in New York. This is an organizing pneumonia. You can see progressive changes. This does not look like ARDS. So if you look at the time course of live virus and laboratory tests. So in green, you see virus isolation. So this is live virus. So as I said before, your viral replication is highest before the onset of symptoms. You're infectious for a few days before to a few days after. And then viral replication goes down. However, you can see the nasopharyngeal swab PCR, which detects viral RNA, stays positive for a long period of time. So the fact that you're PCR positive does not mean you're infectious. The other thing that folks just don't seem to get is the maximal sensitivity of the PCR is about 80% on day 8. 80%. So there are a whole bunch of people who have COVID infection who are PCR negative. This is a tragedy because many hospitals don't recognize this. They think if your PCR is negative, you don't have COVID. That's absolutely not true. So then you see your antibody response, which reflects your cell-mediated immunity, kind of kicks in as the virus starts dying off, and you have simultaneous both IgG and IgM antibodies. So this is really an important slide. So what they did is they did viral culture from the upper respiratory tract. That's where the virus replicates. And then they looked at, was there live virus and the correlation with viral load? So in the dark circles are patients with live virus. So there are two important points. So this is from days of illness, okay, symptom onset. After the 8th day of symptom onset in the study, there was no longer live virus. Those patients who had live virus tended to have the highest viral loads. You can see 10 to the 9 and 10 to the 10. So these are trillions of viruses per milliliter in the nasopharynx. But you can see the PCR stays positive for up to 70 days. Really important. So you can be PCR positive and not infectious. This is a study which shows the same thing. This looked at your cycle threshold, which is the inverse of the viral load, and they showed exactly the same thing. In this study, after day 10, patients no longer had viable live viruses, but remained PCR positive. This is a critical thing to understand. This was an ingenious case trace study done in Taiwan. The question they asked is, how long is someone with COVID infectious? And what they found in the blue line that goes up and down, that after the 6th day of symptoms, patients did not transmit the disease to anybody else. So after 6 days of symptoms, they were unable to transmit the infection. That's why we now use day 10. So after 10 days of symptoms, you're no longer infectious. So how does this virus cause this devastating disease? So as I said, what happens is you develop this profound immune response to viral debris. So you have these viruses that replicate. The host kills the viruses. You have this enormous load of viral RNA, which then stimulates the innate immune response. You have a profound expression of cytokines. It involves the endothelium. You get an endothelialitis. You get activation because of damage to the endothelium, activation of clotting. So you have vascular leak and coagulation. So what this slide shows is that this disease is unlike any other disease we know. The cytokine storm is different to bacterial sepsis, viral sepsis, SARS-1, and MERS. It results in a profound outpouring of a whole host of cytokines. So what they looked at in this study is the relationship between the peak viral load and the peak cytokine level, which may not be on the same day. And what they found is the higher your viral load, i.e. the more virus you have, the more cytokines that you make. And interleukin 7, interleukin 17, interleukin 1 were important, but you can see there's a whole host of cytokines. Now, how does this differ from other respiratory viruses? This is a very devious virus. So if you have influenza, which you can see on the right, what it does is it upregulates interferon and interferon-stimulated genes, and interferon together with natural killer cells are the way the host fights viral infections. And influenza upregulates it, so you have some interferon-stimulated genes and some cytokines. COVID does the complete opposite. It switches off interferon. It's a very devious thing. It switches off the force of the military force that's going to kill it. It inactivates interferon, it inactivates natural killer cell, and then you have this profound cytokine storm. So COVID-19 causes the cytokine storm. So this is a simplified view of an extremely complicated disease that we really don't understand. It's dynamic, it changes, and its complexity actually defies us understanding what's going on. But from this, one can make the inference that trying to block one cytokine or one pathway is kind of absurd because you have all of these redundant pathways and all of these cytokines. So this was an interesting study which they did with SARS-CoV-1. They took pieces of SARS-CoV-1 from SARS-1. They then stimulated mononuclear cells, and what they found is single-stranded RNA fragments. So these are pieces of the virus were found to have powerful immunostimulating activity, causing a profound release of pro-inflammatory cytokines. So this is not the live virus. This is pieces of the viral RNA. And it seemed to signal through toll-like receptor 7 and 8, and inflammasomes are also involved. Once you injected these RNA fragments into mice, they had acute lung injury and they died. So you can see it's not the live virus that's killing the mouse or the human, they're kind of the same, it's the dead virus. In addition to cytokine storm, you have profound dysfunction of cell-mediated immunity, which may be driven by the cytokine storm. So if you look at your T cells are significantly reduced. CD19 cells or B cells, they decreased. Natural killer cells, which are important for viral clearance, are decreased. And not only that, the phenotype of these cells are changed by the virus. This is, as I said, a really devious virus. So it changes it to an inhibitory phenotype with decreased expression of perforin and granzyme that makes the natural killer cells less effective. When you look at your CD4 cells, they decrease, make less interferon. Your cytotoxic lymphocytes are decreased, and they have a senescent phenotype. So you can see it causes profound dysfunction of both innate and adaptive immunity. This is a very good post-mortem study because what they did is they correlated the post-mortem findings with the pre-morbid biochemistry clinical findings in these patients. So this is the pathophysiology of SARS-CoV-2, targeting of endothelial cells, renders a complex disease with thrombotic microangiopathy and an aberrant immune response. So there was endothelial damage and imbalance of both innate and adaptive immune responses, with aberrant macrophage activation playing a central role. And when you actually look at these people who died, they actually have very little virus. There's very little cytopathic effect, and they don't have a lot of viral load. So it again emphasizes why this is such a bizarre disease. It's not the virus that's killing the host, but the host's response to the virus. And this results in an organizing pneumonia, which is important because those pulmonologists or internists who treat this disease as ARDS, you're treating the wrong disease. So this is what it looks like, severe organizing pneumonia with infiltrates of macrophages, monocytes, and neutrophils with this fibrotic intraalveolar process with very scarce virus. As I said, it causes a vascular endotheliitis with thrombosis and angiogenesis. This was a study published in the New England Journal. As we know, there's enormous heterogeneity. Some people have a mild infection, others get severely sick, and others die. So there are a whole host of factors which influence outcome. It seems like inoculum size is really important. So that's why wearing a mask is so critical, because it reduces the inoculum size. It allows, therefore, your host immune response to kill the bacteria. It's also related to ACE2 expression, TMPRSS expression, and then as you'll see, age, sex, obesity, race, immunotype, vitamin D status, and coronavirus cross-reactivity. So there are some common cold coronaviruses which we get exposed to, and they cause an immune response which provides some kind of immunity. This is a study in Annals of Internal Medicine showing the effect of obesity. Obesity is a profound risk factor for death of COVID. It's really important, as is age. So if you're an elderly male who's obese, you're in really big trouble. And you want to lose weight, you can't really change your age or your sex, but you can certainly change your weight. This was a study we did here looking at predictors of outcome. The two most important predictors was age and CRP. CRP is like a summation biomarker of inflammation. There are a bunch of other biomarkers which are really important. The D-dimer, you can see survivors versus non-survivors, lymphocyte count, IL-6, and your serum ferritin. So these are all inflammatory biomarkers which are really important. So how do you manage this disease? So the first thing is you have to understand the disease to treat the disease. And it seems like most people don't understand the disease. So They seem to be floundering in the dark. So the treatment is very much phase specific. So when the virus is replicating, you give an antiviral drug. To give an antiviral drug when the virus is dead is kind of dumb because you can't be more dead than dead. And this becomes important because people are treating the late stages with antivirals. It doesn't work, and in the case of interferon, it's actually harmful. However, once you progress to the pulmonary phase, it's not the virus that's killing the host, it's the host's immune response. So you have to use anti-inflammatory therapy. The treatment depends upon, are you in the symptomatic viral replication phase, or have you progressed to the pulmonary phase? It seems like ivermectin is an antiparasitic drug which has been around for decades. It's very safe, actually seems to be quite effective. As we learned today, and we'll hear of, interferon now is off the slide. Remdesivir is on the slide, but it's going to be off the slide. So you can see this is a dynamic business. The drug of choice for the inflammatory phase is methylprednisolone plus enoxaparin, which is an anticoagulant. So you're dealing with inflammation and clotting. So this is important. So we use oxygen saturation as a marker of lung disease. So in incubation and symptomatic phase, oxygen saturation is stable. As the patient enters the pulmonary phase, their saturation falls. And that's the marker of lung involvement and when you want to start anti-inflammatory therapy. So many patients, for a number of reasons, do not become short of breath. It's a truly remarkable thing. People can have oxygen saturations in the 40s and yet have no symptoms. And we think part of this may be the involvement of your carotid bodies, but also your brain stem, so you're not responding appropriately. That's why symptomatic people at home have to buy a pulse oximeter. You can see as you become hypoxic, it becomes progressive. But as the slide shows, the virus has stopped replicating and the inflammatory response is increasing. So they kind of follow in opposite directions. This is really important to understand the treatment. So we're going to talk about remdesivir. Okay, so this was the first study done in China, randomized controlled trial. The endpoints they looked at were time to resolution or time to get better. There was no difference. When they looked at viral clearance or viral load, there was no difference. This is the final report of the ACTS remdesivir study in the US. What they found is the mean recovery time of 11 days as compared to 15 days. So this is a Wall Street directed endpoint. Most patients want to get better. They don't care if it took 11 days or 13 days. This was a contrived thing to make the Wall Street investors happy. By the way, if you have stock in remdesivir, you want to sell it quickly. So this is the study, the ACT study, showing what I was trying to say. So if you were mildly sick and a little bit of oxygen, it tended to improve time to recovery. However, if you were on high oxygen or non-invasive ventilation, it made no difference, which makes sense because at this point, we don't have viral replication. If you're on a ventilator, it made absolutely no difference. So we can see as you go down this cascade, antiviral therapy seemed to have less benefit. What was very naughty, and most scientists will know this, is halfway during the study, they realized their primary endpoint was not going to be positive, so they changed their primary endpoint to get a positive study. There was the second follow-up study looking at 5 days or 10 days of treatment. It really showed minimal benefit. So I'm now going to present hot data. This was released this morning in Europe. I don't think the results of this study have hit the US yet. When they do, I think we're going to see the stock market react in a not so happy way. So if you have stock in Gilead, you want to quickly sell it. So this is the result of the Solidarity study, which was made available today in Europe. Okay, this is remdesivir. In over 5,000 patients looking at mortality, you can see the two lines overlap each other perfectly. Remdesivir had no effect. And when you looked at subgroup analysis, they couldn't find a group that benefited. So remdesivir failed. Okay, it does not work. We now know that hydroxychloroquine actually failed as well. Patients tended to do worse. We learned today that interferon failed. So unfortunately, folks, we don't have an antiviral that's effective against SARS-CoV-2. The only one may be ivermectin, and we think maybe quercetin. So the antivirals are gone. What about weathering the storm? So we came up with the math protocol. Simple, 1 + 1 = 2. The three core pathological processes leading to multi-organ failure and death in COVID are hyperinflammation, the cytokine storm, the clotting, the low blood levels. So what do? You attack all three. You give a steroid, methylprednisolone, ascorbic acid, thiamine, an anticoagulant. And just for the hell of it, we added a few other little sprinkles on top. That's the full Monty. Why do we use steroids? Because steroids are the most potent anti-inflammatory drugs we have. They act at a whole host of sites. They're non-specific anti-inflammatory drugs. This was an intriguing study. What they did is they took pulmonary alveolar cells, they infected them with SARS-CoV-2. They looked at what genes were up and down regulated. Then using fancy mathematical modeling, they looked at what drugs reverse what SARS-CoV-2 does. They looked at over 2,000 drugs, and the drug which hit number 1 was methylprednisolone. Prednisolone was number 3. So we get to the recovery study, which used dexamethasone in a large group of patients. So remember now we're in the inflammatory pulmonary phase. You can see patients who got oxygen only. There was a mortality reduction of 20%, but the sicker patients on a ventilator, mortality was 35%. So unlike antivirals, which work early, anti-inflammatories work in the pulmonary phase, and the sicker they are, the more the benefit. But what's important is if you're not on oxygen, there was a trend towards harm. You can see this. Those that benefited the most were the sickest, those on oxygen. But if you weren't on oxygen, you did worse with dexamethasone. So we wonder why President Trump got dexamethasone. That's another story. Anyway, so there have been a whole host of studies looking at different steroids. So unfortunately, in recovery, they used the wrong steroid in the wrong dose. So we prefer methylprednisolone, and you can see the number needed to treat to save one life is significantly lower than with dexamethasone, which fits perfectly with the in vitro data. The other problem is you get profound and prolonged inflammation. So if you stop this drug too quickly, you're going to get a rebound. So you've got to treat them for at least 10 days. This is ascorbic acid. That's a goat. A goat makes about 2 to 4 grams a day. When it gets stressed and the stock market goes down or they have a family feud, the goat increases production of vitamin C by about double. Unfortunately, as you'll see, humans and guinea pigs have a mutation in the last rate-limiting enzyme, and humans and guinea pigs can't make vitamin C. So when you stress, you are unable to make this very important stress hormone. So anthropoid primates and guinea pigs cannot make vitamin C. They're the only species on this planet because they have these fatal mutations. Vitamin C actually is a particularly remarkable molecule. It does a whole host of things in sepsis and inflammation. It's a free radical scavenger. It's anti-inflammatory, and particularly as it relates to COVID, it inhibits NETosis and it activates HIF1. HIF1 is really important in changing cellular physiology. It supports lymphocyte proliferation and it stimulates interferon production. So it has multiple actions. This is a study out of China in which they gave patients with severe COVID high-dose vitamin C. you can see the big difference in mortality. Next is anticoagulation for reasons that are unclear. Almost all the professional societies say don't anticoagulate. Why is completely a mystery. So this is a study, again, I think from Mount Sinai, looking at full anticoagulation. So the idea is to be alive and not dead at the end of this. And you can see that the sicker the patients, the greater the benefit from full anticoagulation. So you want to get steroid, you want to get vitamin C, and you want to get heparin. What's really interesting is that anticoagulants like glomerular heparin have a whole bunch of actions. In addition to being an anticoagulant, it appears to specifically inhibit viral entry. It protects the glycocalyx. It prevents netosis. However, as we said, many of these drugs are non-specific, and it's likely in the future we're going to have targeted therapy. What about ventilators? Well, we discovered in New York they treated these patients like they had ARDS. They put them on a ventilator early, and the mortality was 88%. Obviously, that's not good. So what we do is we avoid intubation. We use high-flow nasal cannula. We do what's called cooperative proning and nasal cannula. And the last thing to talk about is the post-COVID syndrome. So this is unlike influenza. People who have COVID don't really recover. About 50% of them have ongoing symptoms. About 50% complain of impaired quality of life. They have fatigue, shortness of breath, joint pain, chest pain. So this disease, even in the recovered patients, they have not recovered. For those who are interested, this is our COVID management protocol on the EVMS website, which goes through prophylaxis and treatment. Just to highlight vitamin D, it seems like NICE thought vitamin D wasn't so nice, but this study shows that the lower your vitamin C level, the greater your risk of getting COVID. And if you get COVID, you have increases your risk of dying from COVID. So giving vitamin D is a very simple way to mitigate against this disease. You hear that anybody talking about this? This is a simple intervention. We then need to talk about the misinformation, because the misinformation epidemic is probably worse than the viral epidemic. And the amount of misinformation is truly staggering. And it's remarkable the World Health Organization has to tell us not to drink bleach because it's dangerous. And it seems that social media is driving a lot of this misinformation. So the bottom line is, don't use social media and just don't perpetuate these falsehoods. In terms of the vaccine, this is what Dr. Fauci said, the chance of the vaccine being highly successful is not great. And I think we put too many eggs in the vaccine basket. So just to finish, the truth has no sides, and we need to speak the truth now than ever before, because that's what informs us and allows us to make rational decisions. Thank you.